Hello, and thank you for attending today's presentation entitled A Novel Method for Enumeration and Speciation of Eimeria Species of Coccidia and Protozoa by Flow Cytometry. My name is Rocío Crespo, and I am a professor at NC State University. Before we move into the presentation, I want to introduce you to my collaborators, Dr. Rafi Kulkarni, Mr. Daniel Adams, who is a student that did most of the bench work for this project, and Mr. Javid Mohammed. Coccidiosis is a gastrointestinal disease caused by parasites of the genus Eimeria. There are different species that affect chickens, and they can be in most part differentiated by the areas of the intestinal tract they parasitized. But the oocysts they produce and are excreted in the feces are difficult to differentiate. There are several available diagnostic methods for Coccidia identification. However, these techniques are labor-intensive, time-consuming, and unreliable, particularly in cases of mixed Eimeria infection. Due to the high prevalence of coccidiosis in poultry, it is imperative to develop a method for a speedy and reliable diagnosis. For these reasons, our objective was to develop a diagnostic method for rapid, automated, and objective simultaneous enumeration and speciation of Coccidia protozoa that affect chickens. We selected flow cytometry. For those of you who are less familiar with flow cytometry, it is a tool to help sort between different cell types easily. We can compare flow cytometry analysis to egg sorting machines. The eggs can be sorted by weight individually, as shown on the photograph on the top. That will be the equivalent of the manual counting, or placed in large machines in the egg plants, where many eggs are automatically and quickly sorted by weight. Similarly, flow cytometry analysis sorts the cells into different categories, which we hypothesized it could identify the different species of Coccidia. We acquire isolates of the individual Eimeria species to optimize instrument settings. The commercial vaccine was used to define the gates where these Eimeria species fall and to test the accuracy of the oocyte counts. This vaccine also served as a positive control in all the flow experiments. Finally, we collected feces from different broiler houses at different ages to evaluate our new method. So let's go and review how we prepare the samples for flotation and flow cytometry. First, we weigh the samples, put them in tubes that have a saturated solution of salt, mix them well, put the mixture through a cheesecloth to remove larger pieces of debris. From here, the samples are placed on the McMaster slide for flotation, and we continue processing for flow cytometry by removing the supernatant after waiting for 5 minutes. The supernatant is placed in a microcentrifuge tube, and we fill the microcentrifuge tube with PBS. Centrifuge for 10 minutes, and we remove the supernatant and collect the pellet. The pellet then was resuspended in PBS and then transferred to a fax tube. This mix was filtered through a 70 micron filter to remove further debris, and then add counting beads. And finally, sent for analysis by flow cytometry. For manual counts, the filtered fluid was dispensed to fill each of the McMaster chambers. The slides were set aside for 5 minutes before they were placed under a microscope and scanned at magnification 4x. To optimize differentiation of oocyst population on the LSR2 flow cytometer, we used 600 microliters from each prepared sample. Each Eimeria species was measured using the height and the width of the forward scatter area and the side scatter area, respectively. The threshold of the forward scatter areas were set at 25,000 to mask about 90% of the debris on the lower end. Concentrations of Eimeria species were determined using counting beads with each field sample, and the stopping gate was set at 5,000 events of the beads. To validate the presence of each Eimeria species within each of the designated gates, populations were sorted using a MoFlow XTP cell sorter. The optimization of the MoFlow settings were done in similar way as we did for the LSR2. Individual Eimeria species were then gated and subsequently collected in separate tubes. The identity of these cell-sorted populations were verified by real-time PCR. We used the QuantiStudio 6-Flex to accomplish this. The sorted populations were tested against known positive samples. Samples were tested in triplicate with both positive and negative controls. Moving on to the results. These graphs all show the same data, analyzed by the LSR2 flow cytometer. We added the color on the graphs to help visualize the different populations. Based on the 4 different colors, we are able to identify 4 distinct populations of Eimeria from a cocktail sample. On the left graph, the data is represented on a dot plot. The middle represents the color contour plot, and the right graph shows the same data on a black-white contour plot. The contour setting on the flow cytometer is a visual aid where the machine tries to predict the individual populations. The circles in the center of the contour lines represent the most certainty of a given population, similar to the contour lines of a topographic map, where the central circles represent the highest point of a given landscape. Our next step was to analyze the same samples with the MoFlow XTP cell sorter and collect each of these sorted populations in separate tubes. The left graph shows the MoFlow results, and the right graphs show the scatter plot image obtained from the LSR2. The results from the MoFlow graph have blue arrows to represent each of the populations. We predicted P1 to be Eimeria mitis, P2 Eimeria cerbulina, P3 tenella, and P4, the largest of the 4, to be Eimeria maxima. The final portion of this experiment was to identify each of those 4 sorted populations by real-time PCR. As you can see, the results in this part of the study were very clear, and P1 was identified as Eimeria mitis, P2 as Eimeria cerbulina, P3 as Eimeria tenella, and P4 as Eimeria maxima. Once we were successful in identifying the populations, we moved to count Coccidia oocysts using flow cytometry. First, we analyzed 3 different vaccine vials at 3 different times by flotation and flow cytometry. For flotation, we counted up to 100 oocysts and classified them into the different species. For flow cytometry, the proportions of each species was calculated using the counts from each species in relation to the total count. There were no significant differences between the proportions found of each oocyst species in vaccine vials, whether they were counted manually or by flow cytometry. To simplify visualization, this table only shows the results from 3 samples taken on 3 different days. Discrepancies were noted between the manual and flow cytometry proportions of Eimeria cerbulina and Eimeria tenella. This is probably the result of similar oocyst size and the subjectivity of the manual counts. Since the proportions were similar, we moved to evaluate the effectiveness of the method with field samples. As with the vaccine, fecal samples were tested by flotation and flow cytometry. Beads were used for flow cytometry to calculate the numbers of oocysts per gram. No interference with Eimeria populations was observed with the use of counting beads. There was a weak correlation between both methods, but all the values fell within the 95% prediction limits as shown on the graph on the left. Overall, the flow cytometry method estimated a higher number of oocysts per gram than the manual method. On the other hand, manual counts had large number of samples with very few oocysts and a few samples with very high numbers of oocysts. This difference may be due to the processing method or the method to calculate oocysts in the sample. Regardless of the difference in oocysts per gram between manual and flow cytometry methods, when we compare the trends of Eimeria populations in a house, they are similar. On the slide, the graphs on the top row represent manual counts, while the bottom row shows the results from flow cytometry. On each graph, the x-axis represents the age of the birds in days, and the y-axis the number of oocysts per gram. A few important points of this study. Sample preparation proved to be the most vital aspect of this study. It is essential to remove most of the unwanted material in the sample. High levels of organic material, similar in size to the oocyst, can cause false increase in oocyst counts. Additionally, too much organic matter can decrease the flow rate of the flow cytometer and in severe cases can clog the machine. Flow cytometry was successful in speciation of 4 Coccidia species from non-cocktail and field samples, without more complex procedures such as antibody labeling or dye staining. On LSR2 instrument, the optimal parameters were found to be forward scattered area and side scattered area, whereas forward scattered width and side scattered area were found to be optimal on MoFlow cell sorter. While the detection and resolution of different Eimeria populations was possible in both instruments, because not all analyzers use the same software, the optimal parameters need to be validated and optimized for each instrument. Counting beads were successfully used to determine concentration of the Eimeria species. No conflict with the Eimeria populations was observed with the use of counting beads. We also demonstrated that flow cytometry is capable of simultaneous enumeration and speciation of Coccidia rapidly and objectively. Although the numbers of oocysts per gram were different by both methods, the proportions of oocysts were similar when using the flow cytometry application and the McMaster method. In general, the flow cytometry method resulted in a higher number of oocysts than when the OPGs were calculated manually. It is possible that debris in the sample interfere with the counts. Both methods proved that the number of Eimeria follow similar trends over time. Overall, flow cytometry counts were more repeatable and had no extreme numbers of specific species. All these observations are encouraging and justify further studies. In conclusion, flow cytometry is capable of speciation Coccidia species and simultaneously enumerate them. The results are encouraging and justify further validation to improve the methodology and agreement of Coccidia counts with traditional methods. Flow cytometry for speciation and enumeration of Coccidia could be used to identify the most prevalent Eimeria species at the farm or region level. It can also help to pinpoint Eimeria species strains resistant to anticoccidials and to improve the selection and production of live attenuated vaccines. Thank you.